And I'm gonna only show my own like ugly face now. All right, so you can hear me, you can see me, you can see the slides.
Yeah.
I think the recording started. Well, let's just get started then. Thank you, Shirley, for the very kind introduction. It's a huge pleasure, a huge honor to be able to speak with you today. This is a very casual discussion. I just want to use this opportunity to tell you a little bit about epigenetics, about DNA methylation. Like Shirley mentioned, I'm Ting Wang from St. Louis, Washington University in St. Louis. Shirley and I have known each other for, wow, how long? Almost 25 years, right? Almost 30 years now. Time flies. So this is my lab's website. I work on epigenetics. If any of you visit my website and read our papers, you're my best friend, okay? All right, so today we're gonna mostly talk about DNA methylation. This is a very brief outline, right? So why we're interested in DNA methylation is really because of epigenetics, right? So DNA methylation is one of the most important mechanism for epigenetic phenomena. So we're going to talk a little bit about epigenetic phenomenon, define DNA methylation, and then we're going to spend some time discussing the normal patterns and functions of DNA methylation, as well as what goes wrong with DNA methylation in diseases, especially cancer, right? DNA methylation is an important mark of the epigenome, and the epigenome sits in between our genome and the environment. We won't be able to talk a whole lot about it, this influence, but I want us to at least understand that the importance of the epigenome, because all the interactions between our genetic material and outside world is mediated through this layer of modification we call the epigenome. And then we're going to spend some time describing some of the technologies that we use to measure DNA methylation. Okay, so we're going to start by going over some very interesting stuff, very interesting examples. These are the classic epigenetic phenomena. So whenever you think about or heard about epigenetics, you almost always come to this example, the agouti mice, right? Look at these two little mice here, these two animals, right? Aren't they so lovely? I mean, they're so lovely that you just want to go and snuggle with them, right? Especially the one on the left, right? The golden fur, very chubby. You just can't stop falling in love with a little animal like that. Unfortunately, this animal has some, kind of bad phenotype, right? So it has high risk of cancer, diabetes, it's obviously obese, and they also have reduced lifespan. So clearly, these two animals, they have very different phenotype, right? They look different, they have different tendencies of developing diseases. But what's interesting of these two animals is they are actually genetically identical, right? So every single base in their genome, from the first base in the first chromosome to the last base in the last chromosome, every single base is identical. Not only that, they live in the same environment, they eat the same food, so everything about their life is the same. So this is the puzzling part, because we've learned so much about genetics. We are in the era of precision medicine, which is all about sequencing our own genomes. We kind of feel that, we're determined by our genes, right? But this is clearly violating that principle, right? So genetically, they're identical, but they have very different phenotype. So what's the difference between these two animals? Turns out the only difference between them are their mother's diet when their mother were pregnant, okay? So the animal on the left, his mother just ate normal mouse chow that we give animals in the mouse house, but the mother of the animal on the right, in addition to normal food, she took vitamin supplements, including folic acid and B12. As we'll mention later, these vitamins actually provide muscle donors, right? So as a result, this animal is more wild type looking, right? So this is a classic example of epigenetics, right? This is also a classic example where people say, we're not what we are, we're what our parents eat, right? So this gives us like very good excuses to blame blame our parents for the stuff that we cannot accomplish in our life, right? So my daughters start to use that on me now. But anyways, so this is a interesting example. But what is the molecular mechanism behind this example? So it turns out in this strain of mouse, right, in their genome, there is a transposable element insertion. So the causal gene is called agouti. So these mice are called agouti mice, right? But about 100 kb upstream from the agouti gene, there is a transposon insertion. The element is called IAP element, right? So this transposable element actually serves as a cryptic promoter for the agouti gene and drives the overexpression of the agouti gene at developmental time and in tissues where this gene should not be expressed. As a result, the animal develop all this bad phenotype, right? But when mothers, right, takes folic acid or B12 as a supplement during a critical window of pregnancy, right, the baby is able to methylate, right, put DNA methylation over this IAP element. When this element is methylated, right, it can no longer act as a cryptic promoter, right? So it can no longer drive the abnormal expression of agouti gene anymore. So it becomes, the gene expression returns to normal, and you ended up with a wild-type looking progeny. So this is the molecular mechanism behind this phenomenon. In other words, the very different phenotype between these two animals, right, are not due to their differences in genetic information. So their DNAs are identical. Rather, this is due to a difference on top of their genome. This is a DNA methylation over the genome. So that's why we call this epigenetics, right? So this is a cute example, but why this is important? So, one thing that we started thinking this a few years ago was, in fact, in our genome, there are a lot of transposable elements like this, right? So in human genome, in mouse genome, close to 50% of the sequences are actually derived from transposable elements. Whereas, only 1.5% of the sequences are protein coding, right? You probably heard about that already. So this makes us think that there's probably a lot of human diseases that are due to kind of the same mechanism. In other words, it had nothing to do with mutations in genes, but rather have something to do with the misregulation or dysregulation of genes, especially when there are transposable elements, right? Usually they're repressed by DNA methylation, but if they lose DNA methylation, they might be able to mess up the normal gene expression. So this is sort of the motivation of a project in a lab where we started to look for agouti genes in human. So in other words, we try to look for human diseases that have, that is associated with highly expressed genes, but these genes are not being transcribed from their own promoter. Rather, they're from a deregulated transposable element nearby. So we set out a study to look for the human agouti gene. So where we spent our effort was in cancer, right? So this is where we have a lot of data. This is where we also know a lot about epigenetic instability and gene expression differences, right? So our reason was this, right? So in cancer, one of the hallmark of cancers is, you have oncogene overexpression, right? So for cancer to grow, you just have to upregulate their oncogene. And that's been studied for 30 years, right? So the major mechanism of oncogene activation has long been studied and written in textbooks. There are three mechanisms: mutation, translocation, and amplification, right? So these are the three major ways for cancer cells to upregulate their oncogenes, right? So we started looking for a fourth mechanism that is epigenetic in nature. So in this case, we're thinking Typically, oncogenes are lowly expressed, transposable elements are methylated, but during tumorigenesis, the epigenome becomes unstable. Sometimes transposable elements become deregulated, demethylated, and start to drive oncogene expression, right? So that logic led to a very interesting discovery. So this is a phenomenon now we call oncoexaptation, which means that oncogenes are highly expressed, right? But this expression does not start from their canonical promoters. Rather, they start from somewhere else. In this case, a deregulated transposable element, right? So if you don't know where to look, you don't find them. But when you know where to look, you actually see this phenomenon is pretty widely spread. In fact, at least 50% of tumors that we looked at have at least one such event happening in them, right? So that's exciting, but what's the usage of this, right? So I'll just go over a very quick example to illustrate that this phenomenon, not only has a prognostic value, but it also tells us, how cancers evolve, right? So let's look at the example at the bottom here, right? So here we're looking at a gene called LIN28B. So this gene is a bona fide oncogene. Again, people have been studying it for decades, and we know that this gene is highly expressed in lots of cancers, especially liver cancer, lung cancer. What people did not know was that this highly expressed oncogene LIN28B usually do not start this transcription from the canonical promoter, but rather they start their transcription from this AlloJB element about 20 kb upstream. This is precisely like that agouti gene example that we found, we talk about all the time in epigenetics, but now we see this is happening in human diseases, in cancer, right? So this transcription start from AlloJB, goes all the way into LIN20B, resulting in high expression of LIN20B. So, if you stratify your patients based on this version of LIN20B, you found that if patients express this AlloJB-driven LIN20B, they feature much, much worse, right? So this is the red line here, right? So it's actually not surprising because they ended up getting higher oncogene expression. So what's the meaning behind this, right? So in addition to providing a prognostic tool, the idea is, we all understand cancer is a process of evolution, right? So you've probably all seen the evolutionary tree people have drawn, to describe like clonal expansion, all this fascinating phenomenon. But so far, all of that work is done on genetics, like following mutations. This actually argues that cancer evolves epigenetically, right? So think about in this case, right? For these cancers to grow, it needs to upregulate the oncogenes. So you can do so with mutations, amplifications, and translocations, but you can also just find another promoter for these oncogenes, right? So in this case, these transposable elements provided these potent sequences that can serve as another alternative promoter, right? So you can imagine this is actually how cancers evolve. Again, this event is not genetics, right? So there's no mutations directly associated with it, right? Rather, this is an epigenetic event. You can imagine that one allele, in this case, what we call an epiallele, had this demethylation change, resulted in a highly expressed oncogene. And now this allele start to take over the population. So it is clonal expansion, but epigenetically, right? So this really changes the way we think about, cancer genome or cancer epigenome evolution, right? So this is an exciting area that we sort of accidentally got into by looking at, transposable elements, probably the most nerdy thing you can imagine. All right, that's like one example, okay? I'm gonna go over a second example just for fun, right? So this is honeybee. We all love honeybee, right? So we all also know honeybee is a social animal. It has, there are queen bees and worker bees, right? So it's kind of different from a human society. So where's the queen bee, right? Well, I kind of put it in right in the middle here for you to look at, right? So you can already see the queen bee looks pretty different from the worker bees. Queen bee obviously also have a very different lifestyle or fate, if you will, right? So queen bees live much longer, right? So they live up to 2 years, I think. And then the workers live a couple of months, right? But the queen bees stay in the hives. All she does is eat and give birth to babies, right? Whereas workers can go out and see the world. So which one's better? Long-lived but never leave the home versus you can go out there, have fun, but you have a short life. I don't know. Your choice, right? Clearly, they have very different phenotype, right? But genetically, they were identical. In fact, when they were born, when we're this little larvae, they're just identical, right? So a few of them were chosen and to be fed with royal jelly. And those few are the ones eventually develop into queens, right? And the rest, you just eat normal food and become workers, right? Everyday workers, right? So, genetically identical, phenotypically very different, right? So this is another example to illustrate what we call this epigenetic phenomenon. So what is the molecular mechanism behind this, right? Turns out DNA methylation plays an important role. So here people measure DNA methylation between queens and workers. You don't have to pay attention to the technology here, but the idea is worker bees are more methylated. Okay, so people did another experiment. They inhibited the enzyme that's responsible for putting on the methylation mark, right? So that the DNA methyltransferase in the workers. So you inhibit that enzyme, and then worker bees' genomes start to become less methylated. And guess what? The worker bees start to grow an ovary. So they start to behave like a queen, right? So this is another phenomenon. Whereas the phenotypic differences is not dependent on differences in genotype, right? Rather, they're dependent on something on top of the genotype, right? So that's epigenetics. All right, so what's in common among this, right? So these two examples, actually, I guess 3 now, right? So the cells share the same genome, or the organisms share the same genome, but they have different epigenome, therefore they have different phenotype. So this is the definition of epigenetics. It's in a mitotically or meiotically heritable state of differential gene activity that is independent of differences in genotype, right? In other words, epigenetic changes influence phenotype without altering genotype, right? So that's kind of a quick definition. Now, there are many molecular mechanisms that enable epigenetics, right? So DNA methylation is the most studied or the most common or most important one, but there are other things like DNA accessibility, histone modification, and some trans factors, right? So today we're focusing on DNA methylation, but before we get into that, I want to mention that epigenetics is not something new. This has been defined for many years. It's actually defined to, it was termed to define development, right? So in fact, epigenetics was used to describe how one cell develop into a multi-cellular organism like us, right? So if you think about it, everybody is actually a walking bag of epigenetics, right? So all the cells in our body more or less share the same genetic information, genetic material. However, they have very different phenotypes, right? Fibroblast cells and neuronal cells, they look very different, but genetically, they're pretty much identical for every single individual, right? So epigenetics was invented, this term was invented to describe this developmental process. And this is described by Conrad Huntington, and then the development is recapitulated by this landscape where at the top of the hill you have a little ball. This represents the topipotent cell that we all started with, right? And as the cell rolled down the hills, at a certain point it's going to make a decision and then branch out and becoming more and more lineage restricted. all the way to the point when they are terminally differentiated, right? So this is what people use to describe differentiation, and in the context of epigenetic potential and epigenetic landscape. This landscape itself is a billion-dollar industry there, right? So people already know how to take a lineage committed cell, or for example, a terminally differentiated cell, and push it back to make it pluripotent, right? And that's very important, have many important implication in medicine. But remember, this process does not involve genetic changes, right? So this process is really an epigenetic change, right? So all we need to do is to restore the epigenome, and all of a sudden the cells will have a very different epigenetic potential, right? So this process is called induced pluripotency. This is not the only, this process is still not very efficient. So people also come up with ways to convert terminally differentiated cells to other cell types, right? So a more direct conversion or transdifferentiation, right? So just a few years ago, I was visiting Gladstone Institute right by UCSF, and they showed me they can grow out fibroblasts. So basically, you can take a skin biopsy, grow out all the fibroblasts, and convert all of them into heart muscles, right? I can see the heart cells beating in a petri dish, and they're actually coming from skin fibroblast. So that's just amazing, right? And that, I can't wait to have that kind of technology around, because I mean, you guys are young, but I've gone through most part of my life, and my heart has been broken many times. Many holes are there. The muscle cells in heart does not regenerate, so I'm waiting for the technology to be ready, take some skin cells, grow heart muscles, and patch all the little holes on my heart, right? So that's the hope. Okay, so I think I'm gonna stop recording, here, and start again. Stop again. All right, so that's kind of the background on epigenetics and some interesting phenomena. So now let's talk about demethylation. What is it? It's actually super boring, kind of simple and boring, right? It's a methyl group, CH3, teeny tiny little group, being put on cytosine, on the C5 position on cytosine, right? So this is catalyzed by DNA methyltransferases. So this is a very simple, kind of boring chemical reaction. It's very teeny tiny group put on the cytosine. In human or invertebrate in general, this methylation usually happens on the CG dinucleotide contact, or CPG. So this is what we usually call CPG, right? So it's so simple. However, by having this methyl group over this C5 position in CPG, the biochemical property of this part of the DNA changes dramatically, right? So without the CPG, without the methyl group, right? So the DNA is like open, happy and being expressed. But with the CPG, the DNA or the chromatin becomes like, condensed, repressed, sad, and, stop, not expressing, right? So biochemically, this is actually putting on a very big difference on the chromatin, right? So that we actually sometimes call methylated cytosine the fifth space. So A, C, G, T, and methyl C, because methyl C has very different biochemical property than the unmethylated cytosine. Okay, so we know a lot about the methylation process. Is categorized by DNA methyltransferase, and then this is sort of a history of how people figured out DNA methylation. So I'm not going to go over this, but if you're interested, there are a lot of literatures to enjoy, right? So for maintaining DNA methylation, or for putting down the DNA methyl, for putting down the methyl group, we rely on two types of DNA methyltransferases. One is called de novo DNA methylation transferases, So we use there are 2 in human, 3A and 3B. They can directly methylate a piece of DNA, right? But we also have another one called DNA methyltransferase 1, which is a maintenance enzyme. So what this one does is during cell cycle, right? So when the replication happens, the newly synthesized strand is not methylated, right? So DNA methyltransferase 1 can actually recognize that and then come in and methylate the newly synthesized strand. So this is how DNA methylation pattern can be faithfully inherited across cell cycle, right? So this is the foundation for this epigenetic inheritance because you can maintain this methylation pattern. So this process has been worked out, many years ago. The reverse reaction, the demethylation reaction, took the field about 20 years to figure out, right? So this is really just figured out over the last 10 years, right? Turns out there are 2 ways for us to remove methylation. One is passive, right? So what I mean is, I just mentioned DNA replication, right? So during cell division, if you make your second strand, right, but somehow you don't maintain it, right? So you don't have methyl groups, you don't have the enzyme, right? So during the cell cycle, if you don't maintain methylation level will reduce by half, right? So if this happens around, for several rounds, very quickly you can lose DNA methylation. So this is how we demethylate our genome through passive mechanism, right? So this is cell cycle associated. But we actually go around and then spend a lot of effort to actively remove DNA methylation. So this actually argues that this must be very, very important because we spend a lot of trouble. So we do this. So there isn't a demethylase, right? Turns out this reaction is very thermodynamically unfavorable, right? So we go through a series of oxidation reaction followed by DNA repair, okay? So it started with methylated cytosine. You use enzyme called TAD. So there's 3 TAD enzymes to oxidate this to hydroxymethylation, to further oxidate to formomethylation and carboxymethylation. And then the cell will recognize formomethylated cytosine and carboxymethylated cytosine and use base pair excision repair system to scoop out the methylated cytosine and then patch in back a unmethylated cytosine, right? So this is sort of the active process of DNA demethylation. So it's pretty involved, which argues, it's probably important, right? Okay, so we're going to quickly go over the normal pattern and function of DNA methylation, right? Turns out CpG methylation is not evenly distributed in the genome, right? So in our genome, as an example, about 70% of CpGs are methylated, and they're mostly focused in or concentrated in the repetitive elements or transposable elements, right? So they're, in fact, their main purpose of DNA methylation is keep those regions repressed, right? So CpG density in the genome is also very, very uneven. So we overall CpG is very, very depleted, except in the CpG island. So these are CpG-rich regions that are usually having important regulatory roles. For example, 70% of gene promoters are actually in CpG island, right? So these regions are often kept unmethylated. Turns out, when we see these are CpG island, CpG rich, it's really, they really have the sort of the expected CpG density. So when when a CpG is methylated, it's actually much easier to mutate, right? So mutation rate on methylated CpG is much higher than unmethylated CpGs, right? So the reason that we see CpG island is because overall, the genome, the CpGs are methylated, so they are mutated away during the course of evolution, except for regions where they're kept unmethylated, right? So these are promoters, regulatory elements, they're kept unmethylated, therefore they don't have an elevated mutation rate, so they show up as CPG island. In fact, if you just look at the CPG distribution in our genome, that reflect methylation pattern of germ cells, right? Because in germ cells, the methylated CPGs start to mutate away, and that's what passed on to in generations, right? So by looking at the just a genome reference, you can actually estimate the methylation level of germ cells. Demethylation has a lot of very important functions, even though this is such a simple modification. The most important, in fact, some people argue that this is the only important function for demethylation is host defense, right? So they're used to silence parasitic sequences like transposable element, like the IAP element that we talked about at the very, very beginning, right? So some people argue that's the only function and everything else is kind of a derivative. But regardless, demethylation plays many, many important roles. For example, in printing, right? In printing is pretty important, interesting, right? We inherit 2 alleles of everything from our parents, right? So that's kind of building redundancy, right? But sometimes, one of the alleles get methylated and does not express, right? So that's a very common phenomenon in placental animals, right? So there are a lot of interesting theories about why we have imprinting. The most dominant or my favorite theory is this is actually how your parents are still having a fight inside you, right? So it turns out, dads are usually very selfish, right? So they only care about themselves and passing on the genetic material. So what they want is big babies. They want their baby to be like really powerful and big, right? So they don't care if the baby takes too much resources from the mother. But moms, they want the babies to be healthy, but she also wants to reserve some resources for herself and the siblings, right? So what do? So mom figure out a way. So mom can methylate the pro-growth genes coming from dads, right? So it doesn't matter how much dads want the baby to grow. Mom said, I'm gonna shut down your pro-growth gene, and I don't care what kind of gene you bring in anymore, right? So that's actually a theory about how imprinting was evolved. So that main mechanism is demethylation. X chromosome inactivation, right? So in human, females have 2 X's, male has 1. Different animals have a different way to balance the expression from X chromosome. What human does is in females during development, right? So this is early stage of development. The X chromosome, the 2 X chromosomes, one of them get randomly chosen and get inactivated through demethylation and long non-coding RNA, right? So as a result, females are actually all mosaics in terms of X chromosome expression pattern, because this happens in early development. All the daughter cells from that point on will express the same X chromosome. So as a result, some, maybe some, over here it's the dad's X chromosome being expressed, but over here is mom's X chromosome being expressed, right? So demethylation is used to establish this X chromosome inactivation. Demethylation is also important for heterochromatin maintenance, chromosome stability, telomere length, and all of this. And also importantly, gene expression control, right? So we're gonna go over some of these in more detail. But before we do that, I want to still describe one of the most fascinating process relating to DNA methylation, that is reprogramming, right? So DNA methylation pattern got reprogrammed twice in our lifetime, right? We kind of know the process pretty well, but we still have a ton that we don't know. We don't know how that's established. We don't know how the specificity is achieved. But let's just go over this interesting pattern. So starting from germ cell formation, right? The first thing the genome does, this germ cell does, is to remove demethylation. So everything gets wiped out. So this is probably how life starts with a clean plate, right? Rather than get everything inherited, we start with a clean plate. So now germ cells will develop, sperm and oocyte. So they establish their very cell type specific demethylation pattern. But when the germ cells, when sperm meets oocyte and form this zygote, right? So the first thing these cells do, right, is to remove demethylation again, right? So this is a second wave of reprogramming, and people believe this is actually how we reestablish this topipotency, right? So this epigenetic potential is reestablished by this reprogramming process. But interestingly, the paternal genome and the maternal genome undergo DNA demethylation in different ways, right? So the paternal genome actually goes through active demethylation. So this is where TAP enzyme is involved. You actively remove methyl groups. But maternal genome goes through this by passive demethylation, which is associated with cell cycle, right? But by the time of pre-implantation, right, the genomes of these cells are all demethylated, except for some imprinted control region. So this is where the previous generation may have some impact on you, right? So after implantation, development start, this demethylation pattern gets faithfully reestablished, right? And then the cell type specificity is often established during this process in development, right? So we know by and large, this is what happened globally, but we don't know the specific mechanisms behind each step and how the specificity is achieved. So there's a lot of interesting stuff that we still want to investigate, right? But in the context of what we want to understand of this normal pattern of demethylation is in relationship to gene expression and genome stability, right? So here I use this to represent a euchromatic region, and over here is this paracentromeric region, right? So the little ticks here represent CPG sites. If there is a lollipop on top of it, this means that CPG site is methylated. Over here, I'm drawing two genes with 2 exons here, 3 exons here. So typically in normal situation, you see higher CPG density in promoters, and these promoter CPGs are usually unmethylated, right? So this actually allows transcription to happen, right? It doesn't guarantee transcription, but this is sort of allowing transcription to happen. So in intergenic region and in gene body, right? So these CPGs are usually methylated, and in intergenic region, you actually see reduced CPG density for reasons that we just talked about. In contrast, paracentromeric regions, these are full of repeats and things like that, right? So these CPGs, these regions have low CPG density compared to CPG island, but the CPGs are all methylated, right? So this is actually really important because these regions have a lot of repetitive sequences, right? So these repetitive sequences are perfect subjects for non-allelic recombination, okay? However, DNA methylation repress that kind of recombination. You can imagine if you lose DNA methylation, these repetitive sequences start to recombine and your genome becomes fragile. This is actually how chromosome get broken, right? So this DNA methylation is important for the stability, okay? All right, I think this is a good place to restart. Okay, so we just went over the kind of the normal pattern of DNA methylation, normal function and pattern of DNA methylation. Let's look at how DNA methylation behave in diseases. We use cancer as an example. Okay, so cancer is a genetic and an epigenetic disease, right? So we all know it's the genetic part, but it's also an epigenetic disease. And there are a ton of crosstalk between these two types of mechanisms, right? So for example, the the two most important hallmark of cancer in the context of demethylation is this genome-wide hypomethylation and increased focal methylation over promoters of tumor suppressor genes, right? So as we just mentioned that when you have genome-wide hypomethylation, the paracentric macroregions are losing demethylation, right? And they're filled with repeats, right? And these repeats are actually perfect subjects for recombination, right? So this might lead to recombination to happen. You're gonna see deletions, amplification, copy number changes, right? So the genome becomes fragile. So this is sort of an epigenetic change leads to a genetic change. Increased methylation over tumor suppressor genes, for example, DNA repair genes, right? Will shut down DNA repair genes, and then cancer cells start to accumulate mutations, right? So this is also a event where epigenetic changes can lead to genetic changes. And vice versa, we also see a lot of genetic changes, mutations in enzymes responsible for chromatin biology get mutated in cancer, right? So by and large, tumor genesis is a process of interaction between genetics and epigenetics. We know this, for a long time. In fact, a lot of the knowledge about demethylation was gained from studying cancer. We know this two-hit hypothesis, right? So this is actually how a lot of tumor suppressor genes are identified. Turns out demethylation is a major, probably the major mechanism for shutting down tumor suppressor genes, right? We know, genes have two copies, right? So in cancer, you need to lose both copies of tumor suppressor genes, right? So sometimes, cancer start with the first copy being lost through a deletion, and then the second copy can be lost by mutation or another deletion. So this is where loss of heterozygosity is defined, or by a demethylation, right? So these are the mechanisms for taking out tumor suppressor genes. So a lot of tumor suppressor genes were actually discovered by identifying this abnormal methylation pattern. So this is a recent example where people show, how epigenetic event alone can lead to tumor genesis, right? So this relates to how chromatin is wrapped around in the nuclei. I believe you will have a lecture on that, right? So here, the chromatins form topologically associated domains separated by domain boundaries, usually bound by CTCF, which is actually a mesosensitive protein. So here, in normal situations, CTCF binding sites are unmethylated, so CTCF can bind here. These two domains are separated, they're doing their own business, they don't talk to each other, right? But in cancer, sometimes the CTCF methylation level goes up, and then CTCF doesn't bind anymore, right? As a result, these two otherwise unrelated domains start to interact. But in the domain on the left, there's a strong enhancer, and in the domain on the right, there's a oncogene, right? So now this enhancer touches this oncogene, resulting oncogene upregulation, and that actually leads to glioma, right? So this is a example where epigenetic event can lead to tumor genesis, and really combines oncogene, demethylation, chromatin domain, all these concepts together. But the typical pattern in cancer, we already actually talked a little bit, is now the tumor suppressor gene promoters are often methylated, right? So again, this is a very common, probably the most common mechanism for cancer to shut down tumor suppressor genes. The other one is this global demethylation, right? So the paracentromeric regions are losing demethylation. So these are the patterns of demethylation in typical cancers, right? So just to summarize, there's a very nice review out there, multiple reviews out there. So in cancer, you see increased methylation over tumor suppressor genes and decreased methylation over paracentromeric heterochromatin. This leads to genome instability. This leads to loss of tumor suppressor gene. That leads to cancer, right? So this is the kind of pattern we see. This pattern actually gives us very important hint, right? So now let's look at these tumor suppressor genes. We know cancer needs to do two things: activate oncogenes, silence tumor suppressor genes. You can silence tumor suppressor gene by mutating it away, deleting it, right? But you can also just methylate its promoter. If you mutated the tumor suppressor gene or deleted the tumor suppressor gene, there's nothing you can do, right? But if the tumor suppressor gene is methylated, the gene is actually still wild type, right? So this actually gives a hope. Maybe this is a new way to treat cancer cells. Well, it's not new anymore. So this is actually a fairly hot field for about 20 years now. People have developed this epigenetic cancer therapy using, for example, DNA methyltransferase inhibitor. You can reduce DNA methylation. Hopefully that also happens on tumor suppressor genes and reactivate tumor suppressor genes, right? So this has been in the clinic for many, many years, 20 plus years. It works somewhat, right? So it works a little bit for blood cancers. In fact, for MDS, this is FDA approved therapy, but it doesn't work for solid tumor. I mean, it's on and off, right? So, the field has gone from, being very, very active to a little bit unsure, but just the past few years, the field sort of is revived for very interesting reason and also related to, transposable element. Right? Turns out, activating tumor suppressor gene is not the only, maybe not even the major mechanism for the therapeutic effect, right? So this type of therapy is not specific. It doesn't target tumor suppressor genes. So it actually acts on the entire genome, right? So as a result, it's not just the tumor suppressor genes potentially getting expressed, the genome getting, the whole genome is getting transcribed. We kind of already said what's the most abundant stuff in the genome. Transposons, repeats, right? So let's say you have repetitive sequences, in antisense orientation, right? Transcription goes through and you ended up getting RNAs that can hybridize and form double-stranded RNA, right? So when the cells sees double-stranded RNA, the cell will think, I got a viral infection. They're going to activate immune response. So people now believe that epigenetic therapy actually primes cancer cells for immune response, right? Now, for patients where the immune system is weak or the cancer has evaded the immune system, epigenetic therapy doesn't work. But for cancers, for patients where you have stronger immune system, maybe it will work, right? So this gives people a hint that maybe we can now combine epigenetic therapy with immunotherapy to improve cancer therapy, right? So there's multiple clinical trials going on to test this hypothesis, okay? I'll just wrap up this part by just telling you, DNA methylation is not just implicated in cancer, it's everywhere. It's implicated in many other diseases. I'm gonna quickly go over these two related but very different diseases that are, we call imprinting diseases. So Angelman syndrome and Prader-Willi syndrome, two very devastating diseases. They're kind of related, but they're, phenotypically, they're very different. These are genetic diseases, so people have mapped where the problem is. The interesting thing is these two diseases got mapped to the exactly same genetic event. They got mapped to the same deletion on chromosome 15. So that's interesting, how the same deletion result in two very different diseases. Turns out, within this 4 megabase region on chromosome 15, there are two imprinted genes, right? One is maternally imprinted, and the other is paternally imprinted, right? So depending the deletion happening on the paternal chromosome versus maternal chromosome, the effect is you're losing different genes, right? Imprinting is interesting, but imprinting also tells you that, we lose this redundancy, right? So one copy is already silenced. It takes just one more hit to render this gene useless, right? So this is why imprinted regions are enriched for human diseases, And this is also why the same deletion, right, have very different effect happening depending on which chromosome it happens on, right? So this is just another anecdote relating DNA methylation with other diseases. Okay, so this is a good good place to stop recording. Okay, so the final section is technology, right? So we're going to talk about how do we detect, how do we measure DNA methylation. This is genomics and bioinformatics. So I'm actually going back to the the old ways of doing genomics. The interesting thing is, we actually, most of the current day genomics is based on old molecular biology and biochemistry, except we have very different ways of reading out the output, right? So this is really how people did genomics back then. I'm telling you this experiment called restriction landmark genome scanning, right? So it turns out that there are restriction enzymes that are sensitive to DNA methylation. For example, NOT1 recognize a motif, CCCC, GGGG, right? But it only cuts when the middle CG is unmethylated. When the middle CG is methylated, it does not cut, right? So taking advantage of this, if you digest the genome with NOT1, if the site is unmethylated, then you ended up with this cutting, right? You can label the end of the DNA chromosome, right? And you can run this DNA on a two-dimensional gel, and you'll be able to see these two dots representing these two fragments, okay? If one allele is unmethylated, but the other allele is methylated, you can you only cut one allele, the other allele is intact, right? You can still label this, and you're going to see the same two dots, but smaller, right? So because you only have the half of the signal. In contrast, if the both alleles are methylated, you lose these two dots, right? So this is the old ways of genomics. You're scanning the genome for regions that are either methylated or unmethylated, right? This is, again, old days. That's how people do genomics. You run this 2D gel. Now you can compare normal and tumor, and you see, hey, I see that digestion happening here, but it's missing tumor. That means there's a methylated site there, right? And then you can actually cut out this piece and then sequence it and then figure out where this piece belongs. This is actually how people identified the majority of tumor suppressor genes, right? So this is a promoter of tumor suppressor gene is methylated. You can find it out this way, right? So this is actually how most of the tumor suppressor genes are identified. They're not actually functionally validated. Most of the tumor suppressor genes in the database that we see do not have functional study. They're identified through this pattern. Okay. So in modern genomics, modern bioinformatics, we're really just taking the old biochemistry, old molecular biology scale up. So I'm going to quickly go over 4 types of technologies for DNA methylation: enrichment-based, restriction enzyme-based, bisulfate-based, and the future sort of a direct long reads, direct reading based, right? So first, enrichment-based. This is actually very, very simple, right? So this is just like ChIP-seq, except easier, because you don't even handle chromatin, you handle genomic DNA, which is much more steady, stable than chromatin, right? So here you rely on enzymes that or antibodies that recognize methylated cytosine, right? For example, antibodies recognize this cytosine, and you use, you do a chromatin, basically you do a immunoprecipitation, right? Take the genome, sonicate, use antibody to pull down methylated pieces, and you can sequence them, right? So the representative technologies are MEDAP-seq that uses an antibody, and the MBD-seq, this actually uses a methyl binding protein, and then pull that binding protein down to enrich for methylated region. So if you get that data and then put it on a genome browser, they just look like a typical ChIP-seq data, right? So this represents methylated regions, right? So here is one gene, promoter, CpG island. If you look at the methylated regions of the MEDAP data, you see those little peaks representing methylated fragment, right? So this is a genome-wide approach but the resolution is kind of low. You put down fragments, but you don't necessarily know which exact CpG is methylated. Okay. You can also take advantage of restriction enzyme, right? Just like the first experiment I mentioned, rather than running a 2D job, you can sequence them, right? So that's what's behind this so-called MRE, or methylation restriction enzyme based method, right? So again, you have unmethylated cytosine in a restriction enzyme recognition site, and a methylated cytosine. In this case, HPA2 recognizes CCGG, but it only cuts when the middle CG is unmethylated, right? So you're gonna cut here, but not here. And then you can sequence now the end of these sites. Now you know precisely where the unmethylated CpGs are located, right? So this is MRE-seq, right? So typically you can plot this type of data on a genome browser and then see this little ticks represents the number of cut happening on those specific unmethylated CpGs, right? So you can see, promoters are unmethylated, right? And this is a 3' CpG island that's partially methylated, right? So these two types of data are sort of complementary to each other. One enriched for methylated region, the other enriches for unmethylated region, right? So you can also use, the limit here is like recognition site, right? So not all CpGs are within recognition site of a restriction enzyme. So you can use multiple enzymes to increase this, this coverage, right? So this is also a typical technology. But the gold standard, right? The gold standard is bisulfite sequencing, right? So, so the two methods I told you earlier are enrichment based, which means it does not tell you the absolute level of methylation. You get the relative enrichment. But bisulfite sequencing gives you the absolute level. It's a digital readout, right? So bisulfite is a treatment that can turn unmethylated cytosine into uracil, right? So in this process, methylated cytosine is protected, so it's not being converted. But unmethylated cytosine got converted into uracil at a very high efficiency, like usually 99.5% of the time, right? So when you sequence this cytosine will show up, this uracil shows up as a T, right? So when you see a T, your interpretation is, that cytosine was unmethylated. But if you see a C, you know that cytosine is unmethylated. So you get this digital output of methylation level. So all the unmethylated cytosines are being converted, not just in the CG context, right? So there are a lot of Cs not in CG context. They are actually also getting converted. As a result, your library, the library that you sequence, or in the end your reads are depleted of Cs, right? So so it creates some trouble in bioinformatics like mapping, right? But those problems have long been solved, right? So typically, there are multiple packages out there. So this is a typical pipeline for analyzing whole genome bisulfite data. I believe you might have a homework on this. So you align your rates to the genome, and then just count the regions where C and T's got CNT ratios, right? So then you can figure out the methylation level going from 100% methylated to 0% methylated, right? So, and then you can do your differential methylation analysis and so on and so forth. So many packages have been developed to complete this process. So whole genome bisulfite is a gold standard, but it's not without limitation. One of the key limitation is you have to sequence the genome many, many times, right, in order to get this this ratio, right? The standard is usually you have to sequence the genome like 10 times, 20 times, up to 30 times to have an accurate methylation call. So as a result, it's pretty expensive, right? So you have to, every time you do this, you're resequencing genome. We also already know that CpGs are, not evenly distributed in the genome. There are regions where CpG densities are high, regions CpG densities are low, right? When you do a whole genome bisulfite sequencing, you're sequencing everything. as a result, you can look at your own data, but 70% of your reads don't even have CpG on in them, right? So you're actually wasting a ton of data and money with this type of with this approach. So one way to combat this is through a protocol called reduced representation bisulfite sequencing. You're still doing bisulfite sequencing, but you try to enrich regions where there are a lot of CpGs, right? So what you do is you digest your genome using enzymes that recognize high CG content motifs, like MSP1 recognize CCGG, right? So get this fragment and you only select a specific range of fragment size, right? So these regions with low CpG density get thrown out, and you focus on regions with high CpG density. These are usually the CpG island and promoters. So as a result, you don't sequence the whole genome, but you sequence a fraction of the genome, but your per CpG cost is going to be much, much better, right? So this is the advantage of reduced representation bisulfite sequencing. So further, you can further reduce that by using arrays, right? So Illumina actually made arrays for methylation. The concept behind this is actually genotyping, right? So think about this. Now you converted your cytosine into T versus not as a function of methylation, right? So you can genotype the specific cytosine, right? If you know what CpG you want to look at, you can use an array to genotype it, right? So that's precisely what people did with this Infineon array, right? So you interrogate the very specific CpGs you want to know. You simply ask, is it a C or a T? And then you can get the methylation level, right? So this is a very commonly used approach. The advantage here is it's very reproducible, right? And you know exactly what CpG you look at. The disadvantage is you only know what CpGs that you interrogate, right? So there has been like 4 generations already. The first generation is even smaller than this. I think it's like 1.5 thousand. And then there's this 27,000 array, 450K array is the most widely used one. Most of the data in the public domain are this 450K array data. Recently, people switched to 850K array to get an even better coverage. But how many CpGs are there in the human genome? There are 28 million, right? So you're not getting all of them with this array. The other thing is, so far commercially, just human array is available. They've made a mouse array. It took years, but finally they made one. I think it's going to become available very soon, if not already, right? But again, if you have a new species or interesting species you want to go after, you don't have an array, right? So you go for whole genome bisulfate or RBS or other technologies. So finally, I'm going to wrap up and then just tell you the newest technology that's out there, right? And we have a lot of hope to directly read out modified base. You don't enrich, you don't modify, you don't change, you just read it out, right? And that relies on the current 3rd generation sequencing technology being even better, right? So the 2 dominant forces in the field is PacBio, SmartSeq, and Nanopore, right? In fact, 12 years ago, when PacBio just started getting into the market, they already said, hey, we should be able to read out modified base directly, right? So PacBio uses this, measures this kinetics of DNA polymerase incorporation, right? So when the base is modified, this kinetics changes a little bit, right? So they wanted to capture that, right? So they've been working on this for over a decade, but the quality has always been suboptimal, right? Until recently, right? So this paper, I think, just came out maybe a month ago. That kind of, my prediction is it's going to change the game, right? So they used a neural network to train not a single base anymore, but a window of bases to improve the accuracy of calling modified base methylated cytosine, right? So finally now after 12 years, I think, they're now probably in real business to be able to read out DNAMethylation directly, right? So that means long reads and DNAMethylation, it can't get better than that, right? So nanopore sequencing came a little bit later than PacBio, but in terms of DNAMethylation, they actually did better, at least in the past a few years. So they, this DNA strand goes through this nanopore, and depending on the size of the base, you can measure the change of electricity, right? And then you measure the change of this current to figure out what basis it is, right? And then the modified base have a different size, so that's how you tell that apart. The reason that they are doing better, I think, is because they already use this window-based approach to call base, right? Not a base at a time, but a window at a time. So the same base actually get caught multiple times, in the same neighborhood. So that gives higher accuracy. In fact, last year, this telomere to telomere a genome paper, and now they have a second paper in bioRxiv already have data on this nanopore-based DNAMethylation estimate for some interesting like Central America region. So this is a super exciting direction, because those regions, not only we don't know their DNAMethylation, we don't even know how those sequences look like, right? So now we have long-read technology that allows us to go through this, hard to measure regions, and then be able to also reveal their epigenetic pattern or their DNAMethylation pattern. Okay, just finally to wrap up, I want to introduce you to some of the resources out there. We helped build the human reference epigenome. So this is a collection of data from the Roadmap Epigenomics project, which ended a few years ago, but other consortium projects like International Human Epigenome Consortium, ENCODE, 40N, Target, all these projects are continuing adding data to the collection, right? So we built a genome browser, and we are now hosting over 200,000 genome-wide epigenomics data now, including DNAMethylation, histone modification, and many different data sets. So there is a resource like this, and there are indeed many resources where you can get this data for free and then just have fun. And the last resource I want to mention is this upcoming resource, right? So this is a new project, Human Pangenome Project. So this is a project where we're reinventing the human reference genome. The reference genome everyone uses, a map data tool, is very useful, but has its own limitation. It has places missing, and it's also, whose genome is it, right? So it's actually nobody's genome. It does not represent the human diversity. So a couple of years ago, this new consortium got started, to build new human reference genome that incorporate more diversity, have better representation. So this is an ongoing project, and we are also epigenetically annotate these new genome references. So there are a lot of upcoming resources for those who are interested in. I think that wraps up everything I have prepared. I'm going to go ahead and then stop sharing here, stop the recording here.